Hi, I am Madhurima Datta, Zoology and Biology Educational Content Creators. Today's content for Class 12, CBSE, ICSE, other boards and also NEET aspirant students. Today's unit is Reproduction of Plants and chapter Sexual Reproduction in Plants and topic is Flower Structure. Let's start. Plants reproduce asexually and sexually both types. The propagules through which the plants reproduce are the leaves, roots, stem, while the reproductive organs for sexual reproduction is the flower. All flowering plants show sexual reproduction. A look at the diversity of structure of the inflorescence, flowers and floral plots show an amazing range of adaptation to ensure formation of the end products of sexual reproduction, the fruits and seeds. Next, flower a fascinating organ of angiosperms. Uses of flowers. Flowers have been of great importance to humans. It has aesthetic, ornamental, social, culture, and religious values. They have always been used as symbol for conveying important human feelings such as love, affections, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at home and in the gardens. Next, structures of the flower. Flowers are structurally divided into the following different wholes. First, calyx, group of sepals usually green in color. Next, corolla, group of petals brightly colored except green, which attracts the bird and insects. Androecium. Androecium is a male reproductive part of a flower consisting of the stamen having filament and anther. Next, gynoecium. A female reproductive part of a flower known as stele or carpel consisting of stigma, style and ovary. Next, there are many structural and hormonal changes that take place in the formation of the flower. These changes leads to the modification of the floral primordium and thus the arrangement of the different wholes of the flower. Next, sexual reproduction in flowering plants has been divided into the following events. First, fertilization, structure and events. Next, fertilization event. And last is the post-fertilization structure and events. At first, we will know about the pre-fertilization structure and events. First, structure of stamen. Stamen consists of two parts, filament and anther. Filament, it is a sterile part which connects the anther to the thalamus or petals of the flower. Next, anther. It is the fertile part within which are formed the pollen grains. Anther is usually bilobed. Each lobe has two theca and thus known as the diethiocells. Next, each theca has two microsporangia, 4 in total, and thus a typical angiosperm anther is tetrasporangiate. Each microsporangium develops and becomes the pollen sac that produces a large number of microspore known as pollen grains. Next, anther development. Anther is a parenchymatous in nature. Hypodermal archosporial cells are differentiated one in each angle. Each anthosporial cell undergoes a periclinial division to form an outer primary partial cells and inner primary sporogenous cells. The primary parietal cells undergoes division to form 3 to 5 layered anther walls. Next, wall layers of the anther development. First is the epidermis. It is the outermost wall layer of the anther. It is the protective function. Next, endothecium. The endothelial cells develop a fibrous thickening before the dehiscence of the anther. It occurs in the form of radial bands on the targeting wall. This thickening is made up of a cellulose with a little pectin and lignin in some cases. It is a hygroscopic in nature. It helps in the dehiscence of the anther and dispersal of the pollen grains. Next, middle layers. Middle layers may be range of 1 to 6 or in some cases several middle layers are present. Middle layers are degenerate at maturity of the anther. They provide nutrition and also help in the formation of the pollen kit. Next, tapetum. It is the innermost layer of the anther, provides nourishment to developing pollen grains. The tapetum is of two types, amoeboid or periplasmodial or invasive. Tapetal cells break up, releasing the protoplast into the pollen chamber. All such protoplasts now fuse to form periplasmodium. Microspore mother cells get surrounded by periplasmodium. Next, secretory or glandular or partial. This type of tapetal cells remain throughout the development of microspores and finally they degenerate. Today's topic has ended. Next day we know about the microsporangia and development of pollen grains.